Sorry, I got a phone call and my video stops when a phone call comes in, so I apologize. So we have Dolly the sheep. She's a lamb cloned from an adult sheep by nuclear transplantation from a differentiated mammary cell. So Dolly's premature death in 2003, as well as her arthritis, led to speculation that her cells were older than those of a normal sheep, possibly reflecting incomplete reprogramming of the original transplanted nucleus. So here, let's look at what happened. So what they did is, this is Dolly's, this is the donor. So the mammary cell donor. So this is, they took and cultured some of her mammary cells from her mammary glands in the laboratory. So there's the cultural mammary cells. It says are semi-starved, arresting the cell cycle. So what happens is they stop dividing, and it causes de-differentiation. So starving them, they found they kind of make them less differentiated, because these were mammary cells. All right, so here's that donor cell. And then at the same time, they were working on getting the egg cell donor. So they took an egg cell from another sheep, so the egg cell donor, and removed the nucleus. So that's where they're actually sticking kind of like a little kind of very micro pipette to go into the cell. So they have to do this underneath the microscope and get the micro pipette, and they're showing you here the nucleus is removed. So here's a little micro pipette. This is the nucleus right there, and they're sucking it out. And so therefore you have an egg cell with no nucleus. So then they fuse the cells. So they fuse one of these mammary cells, the donor cell, and each one of these mammary cells has a nucleus that's diploid, and then this egg cell no longer has a nucleus. So they fuse them together, and what you now have is what is like a fertilized egg cell. You have what was the egg cell, but now it has a diploid. This is a diploid nucleus from that mammary cell. So this has all the DNA that's genetically identical to the mammary cell donor. So then they grow it in the culture and allow it to divide. So you get an early embryo here. And then they take that embryo and implant it says, in the uterus of a third sheep. So it's not even one of these guys here. So implant it into the uterus of the third sheep. And so this is the surrogate mother, because it's not really her baby. It's not her DNA. It's this one's egg cell and this one's DNA. And so therefore this one then will carry, and hopefully the thing is that the implanted embryo has to attach to the uterus and to continue to develop and so on into a lamb. So here's Dolly, the first one ever to happen to do this. But what people don't realize is that it took hundreds of tries to get Dolly. So most of the time, hundreds of times, this didn't work. They tried to put it into the surrogate mother, and it would not. It didn't take. And so therefore, it took a long time to get Dolly, and so this is why it was a really big deal. So Dolly is genetically identical to the mammary cell donor up here. So they're actually the same genetically. It doesn't necessarily mean that they'll behave differently. And so they found cloned animals. They've cloned cows and so on, and they have different behaviors. Some are more dominant, some are more submissive in behavior. And so they found a lot of variations, even though they may be genetically identical. And so this little baby would be genetically identical to this one. So a lot of people think cloning, we can make somebody exactly like, Johnny over here. But you can't get cloning from an adult to another adult. What would happen is, like if we relate this to like human cloning, let's use this as an example. So you have, let's say you have Susie here, she's the egg donor, and you have, I don't know, let's say Maggie over here. Okay, so Susie donates the egg, Maggie donates the cell that you're going to take the nucleus from, all right, and so then you get rid of the nucleus out of Susie's egg cell, you fuse Maggie's somatic cell with it, and so now you have the nucleus, but really this is Maggie's nucleus, and so therefore then it begins to divide. You put it in yet another individual, all right, let's say Tracy, and you put another individual and implant it into their uterus, and then you get the baby. So the baby is genetically identical to Maggie. All right, and so therefore Maggie is still an adult. Her clone is now a baby. All right, so when you're cloning, the clone starts out in life as a baby, is what I'm trying to say, and has to grow up and will be genetically identical to Maggie. All right. And so that's how cloning works. And so in 1997, cloning has been demonstrated in many, or since 1997, cloning has been demonstrated in many mammals, including they've cloned mice, cats, cows, horses, and pigs, lots of different organisms that are cloned using this method. The thing is, let me just go back to Dolly. The thing is that Dolly didn't last very long. So she says the premature death in 2003. She actually gets a lung disease that often is common in older sheep, and so she died. She actually had a lot of things wrong with her that usually were wrong with older sheep. So even though while she was young, in terms of her birth on the earth, her cells were behaving as if they were older, and she had some older characteristics. So some of those things, remember, like whenever the cell divides, you get the shortening of the chromosomes, the telomeres at the end. So you've got all these things going on. So you've actually given older DNA from an older cell to a brand new baby, and so the baby has some characteristics of older individuals. So that's a real problem with cloning, is the side effects from that. All right, so let's flip it over. Copycat. I love this name. Copycat was the first cat cloned, and let me show you Copycat. Here's Copycat. Look at how cute. So Copycat is a cloned cat, obviously. And so that's just another example of an animal that's cloned. So let's look at the problems associated with it. In most nuclear transplantation studies, only a small percentage of cloned embryos have developed normally to birth. So I said it took hundreds of times to get Dolly. Many epigenetic changes, such as acetylation of histones or methylation of DNA, must be reversed in the nucleus from a donor animal in order for genes to be expressed or repressed appropriately for early stages of development. So as these cells differentiate, and you take a differentiated cell from an older individual, some of those genes in that cell has been turned on, some of them have been turned off to make that cell a mammary cell. And so we have to kind of undo that. So remember, acetylation of histones and methylation of DNA, this turns genes off. And this one turns it on. So certain genes are on and off in there. So you have to kind of reprogram it so that those genes aren't turned on and off, because in an early embryo cell, it's not differentiated, and so we don't want it to behave as a differentiated cell. And so those are some of the problems associated with cloning animals with a nucleus from a cell that is much older. So what we can do is use stem cells. So let's say the stem cells of animals. A stem cell is relatively unspecialized that can reproduce itself indefinitely and differentiate into specialized cells of one or more types. So some cells isolated from early embryos at the blastocyst stage, so this is very early on in development, are called embryonic stem cells. The adult body also has stem cells, which replace non-reproductive specialized cells. Embryonic cells are, there's that word again, totipotent, meaning that they can differentiate into all cell types, whereas adult stem cells are what we call pluripotent, able to give rise to multiple but not all stem cell or all cell types. So if you look here, there's a lot. This is a very controversial topic in terms of doing research on stem cells. Let's look at the difference here, embryonic versus adult stem cells. So embryonic stem cells, as it says here, you can take most of the cells and very early on in development are stem cells, which means they have the ability to become any cell in your body. So these are totipotent. And so we can take stem cells and we can culture them in the laboratory, and depending upon the different environments and what chemicals we give them to trigger turning on or off certain genes, those embryonic stem cells can be triggered to differentiate into all types of different cells, so liver cells, nerve cells, blood cells, all right, and so on. And so embryonic stem cells have the ability to become any type of cell. Adult stem cells have limitations. And so adult stem cells, these are stem cells in your bone marrow. This is an example of that. And what this picture is trying to show. So you take these cells out. The stem cells in your bone marrow can become all different types of cells or blood cells. And so they can become white blood cells, red blood cells. So that's what this is supposed to be showing you here. So this is RBC. This is a white blood cell, and so on. So these are stem cells, meaning that they can differentiate and be triggered to differentiate into different types of cells, but they're limited. These are different types of blood cells. So these cells from the bone marrow can't be triggered to become a nerve cell or a liver cell. It can only become one type of blood cell, whereas the embryonic stem cells can become all types of cells. And so therefore, that's the difference between them. And so you can see that stem cells, you can create in the laboratory using stem cells many other types of cells that can be useful to help other people and do research on. So for instance, there's another place besides an embryo. There are stem cells in the umbilical cord that are totipotent as well. And so you can, when a baby is born, you can choose to isolate those stem cells out of the umbilical cord, and they can actually freeze them. And then what happens is they're doing research on a lot of different diseases and so on. One of those diseases is like type 1 diabetes, where you have pancreas that the cells do not produce insulin, so they're doing research to make cells that will make the insulin, so normal pancreatic cells, and so on. And so there's research being done on that. But I just want to point out that with the embryonic stem cells, there are also cord blood stem cells as well. And so like I said, a lot of promise in terms of research done on this, but also controversial, as I said, especially the embryonic stem cells, about whether or not that's right or not, and because what happens when you use the stem cells is the embryo actually dies, and so you can see where it can be very controversial in terms of whether or not we should be doing that at all or not. All right, and that is the end. So we will review tomorrow.